Hi, my name is Kevin. I'm a PhD student at the University of Toronto in Dr. Yuri Raymond's lab. My project is about treatment-associated mutations in cancer. I will talk about how we measure their frequency in regulatory elements and try to understand their functional implications with computational biology methods. Just to give a little bit of background, most mutations in cancer genomes are selectively neutral mutations. They are called passenger mutations. Although they are not main drivers of carcinogenesis, they reflect the mutational processes that contributed to tumor evolution, and they have the potential to serve as biomarkers. So we're interested in studying them. And importantly, passenger mutation rate is variable throughout cancer genomes. And a major covariate of the mutation rate is the presence of regulatory elements. One example is the binding site of the CTCF protein, which is the central regulator of chromosome organization and gene expression. These binding sites have a particularly high mutation rate in many cancer types. Cancer treatments are often mutagenic and leave mutations in cells that survive the therapy. So treatment-induced mutations contribute to the catalog of passenger mutations when those cells propagate as recurrent or metastatic tumors. And in this project, I'm interested in the contribution of cancer treatments to the mutational landscape of regulatory elements. My hypothesis is that because transcription factor binding sites are more susceptible to mutations than other genomic regions, treatment-induced mutations affect them more severely. This could be reflected by firstly a higher mutation rate associated with treatment, and secondly a change of mutational processes associated with treatments. My second hypothesis is that mutations in regulatory elements have a high chance of causing functional disruption. This is because binding of transcription factors often depends on specific DNA sequences, so mutations disrupting these binding motifs may deregulate gene expression. Our data comes from the Heart Medical Foundation, which has a catalog of about 3,600 metastatic tumor samples. These patients have gone through various treatments prior to tumor sampling, and this data set is very suitable for our study. So to measure the mutation rate variation of a class of elements in general, our lab has previously developed a statistical method called RM2. This method takes a set of regulatory sites, define it as the same sizes, and align them and analyze their mutation rates together. And it uses the flanking regions of each site as control to calculate a background mutation rate for comparison. And each site may have different trinucleotide sequences and local mutation rate, and these factors are all taken into account in the Poisson regression model. So I first focused on analyzing a group of around 32,000 CTCF sites that are constitutively active, meaning that they show CTCF binding across most tissue types. With RM2, I measured mutation rate changes in CTCF sites and their association with different treatment types in any available cancer types. From a screen of 89 groups, we found 4 associations in total between different drugs and mutation rate in different primary tumor types. Among them, we verified that radiotherapy is associated with the highest mutation rate increase in CTCF sites, specifically in metastatic colorectal cancer. There were 3 other associations that were statistically significant, but have weaker signal, so we focused on characterizing the radiotherapy association. My second aim is to decipher the mutational processes in CTCF sites and those associated with radiotherapy. This is done by decomposing the catalog of mutations from all samples into characteristic trinucleotide profiles associated with known mutational processes. Then I assigned each mutations to their most probable signature, and this allows me to compare mutational signatures across genomic regions and sample groups. Here shows the proportion of signatures in radiotherapy treated and untreated groups in CTCF sites and in other regions. Across the board, the 2 treatment groups and different genomic regions all showed mutational signatures common to colorectal cancer. However, the proportion of signatures is broadly different in the radiotherapy exposed samples. Most visibly is the increase of SBS17 and SBS5 in radiotherapy treated samples. This increase is variable in different regions of the genome, but CTCF sites appear to have the particularly high increase associated with radiotherapy exposure. We verified this by measuring mutation rate again with RM2, but on individual signatures. We found that despite DNA sequence and background mutation rate difference, SBS5 showed a higher activity in CTCF sites when tumors are exposed to radiotherapy, whereas signatures 17A and 17B, they showed an increase in both CTCF sites and in the flanking regions. So this suggests that radiotherapy may affect at least a subset of mutational processes differentially in CTCF sites. After establishing the high mutation rate and mutational signatures in general, we also want to know whether there are individual CTCF sites that are particularly vulnerable to mutations, or that they have been selected for functional disruptions. We looked for CTCF sites with unexpectedly high mutation rate with our driver Discovery 2 active driver. It works by establishing an expected mutation rate from the surrounding 100 kilobases of an element and comparing that to the observed mutation rate of that particular element. With this, we found about 200 CTCF sites with an aberrantly high mutation rate. Using a simple initial approach, we associated the sites with their nearest genes. And interestingly, we found that the top mutated CTCF sites are all located around known cancer-associated genes. We also performed pathway enrichment analysis with active pathways, and again we're associating CTCF sites with their nearest genes, and active pathways ranked the genes by mutational enrichment and compared with known pathways. Interestingly, highly mutated CTCF sites converge near some extracellular matrix pathway genes, which are related to metastasis. To summarize, with computational biology approach, we found that radiotherapy is associated with high mutation rate and altered signature profile in CTCF binding sites, specifically in metastatic colorectal cancer. We also found evidence that the highly mutated CTCF sites may be associated with known cancer genes. And overall, our results suggest a potential side effect of radiotherapy through non-coding mutations, and it provides motivation for more detailed study. Thank you for listening.